Yep, and we are back. Second hour here, Friday. Now starting metabolism after we finish off cholesterol here and hormones. We'll do a little bit on hormones, HDLs, LDLs. I mentioned the statin drugs inhibiting HMG-CoA reductase. Keep in mind what's due here today in review. We'll review both for test 4 and the final on Monday. We should have time there. But be looking at those sample test 4 and the sample final that we have there. Yeah. And so, yeah, test 4 will have a little bit on metabolism. But again, I'll say this a couple times. I'm not expecting you to memorize the whole pathway and reproduce the whole 10 and then 8 steps. That's 18 more reactions. No, I'm expecting you to look at a reaction in isolation and decide what it is. Either I'll give you the enzyme and all the conditions, the cofactors, and then what is the product? Or I'll show you the product and ask you something about what the enzyme may have done. But you should be able to figure out all of them by inspecting the structure and thinking about that using the principles that we've covered here. And then any mechanism, my favorites are the NADH and the NAD plus ones, transferring hydride or getting a hydride off a substrate. And you'll see that again here in metabolism in a couple spots. And yeah, if you master those cofactors, you're really going to have a leg up, I think, in 481 going on in biochem. All right, let's finish off cholesterol here. We looked at the most amazing enzymatic reaction ever. Squalene oxide to lanosterol. It took many people working on it. I think there were 3 different Nobel Prizes awarded for cholesterol biosynthesis. A lot of fundamental work here, going from squalene to squalene oxide, and then how it gets to lanosterol. This part I'm not going over. We'll leave that as a mystery. It is quite well known how it's transformed lanosterol into cholesterol. But that's the actual interstitial membrane component, right? Up to 20% of the molecules in your cell membranes are cholesterol. The hydroxyl group is positioned toward the outside, toward the phosphatidyl zwitterine part, and then the hydrocarbon part sticks down in the diester, the fatty acid area. And it breaks up that packing forces between those straight chain groups. Those straight chain groups, remember, are saturated fats. So at room temperature, they should be what? Solids, right? But cholesterol is added to keep that fluid. In fact, the membrane model is called the fluid mosaic model. You've probably heard that term before. Anyway, let's look at that mechanism again in a little detail. Yeah, I did cover this. I was just going to review it briefly. But yeah, know how to, think about how the enzyme works here, protonating the epoxide. That's going to open it up, right? And then the cascade, the flow to form the rings, and then the shifts here, okay? So we could ask you about little basics of that, but I did tell you I've never put this on a test before, so know enough about it, look through it enough to appreciate the fundamentals. And this as well, I don't expect you to know much here except that cholesterol is what? Not a hormone. Cholesterol is the precursor to all the hormones. Here's the basics of it in the body, right? These are all steroids in that they have the 4-carbon ring, the 4 rings, and then different functional groups on them. In the body, cholesterol is first converted into progesterone in the adrenal glands and the ovaries. Only a very small amount, though. This helps control pregnancy effects in females. Adrenal glands for males, yes, guys, you have some progesterone. This idea if they're all one male or one female is incorrect, actually. There's a cascade relationship here synthetically of how they arise, which I think is interesting. Progesterone goes to testosterone then, the classic male hormone, to develop muscle mass and vocal cords and hair growth, whatever. But yeah, it has an enone in the A ring, and I expect you to know that. The basics of it, the secondary alcohol in the A ring here for cholesterol. Don't worry about drawing the whole structure, except know it's a hydrocarbon, right? Testosterone has the chain taken off the D ring. It's now an alcohol. It's more water soluble, okay? It can diffuse in the body. So it's an enone here and an alcohol there. And then estradiol and estrogen in the ovaries control female effects there. Does the aromatization of the A ring. And there are drugs that affect that pathway. That's an important therapeutic pathway. aromatases, they're called. But yeah, know the basics of that. The male hormone, enone, female hormone, phenol. And that's it there. There's also the cortisones, the cortisols. You've probably heard of some of those that control inflammation. Also metabolism, glycogen, um, immune bone growth, cortisol, androstenone, blood pressure, electrolytes. There's some different hormones that have different effects. The adrenal glands tend to be very dominant there. Remember, the adrenals also make epinephrine and norepinephrine, those two fight or flight neurotransmitters. Then cholic acid, this is an interesting one. This is in bile salts, so the gallbladder makes this. It's an emulsifier for the digestive tract. Why do we say an emulsifier? Well, it's highly oxidized compared to cholesterol. In fact, it's a salt, a sodium salt, carboxylate salt. So it has a very polar side to it, and then a somewhat nonpolar thing here. So it can actually function like soap in the body and emulsify different dietary fats. Now, if your gallbladder's not working right, you can have all sorts of digestive problems. So you have to lower the amount of fats in your diet or take supplements there. So cholic acid has the function of emulsifying or softening stool materials in the GI tract, which also comes from cholesterol, but highly functionalized, right? A couple more alcohols and a sodium salt there to affect how it works as a bile salt emulsifier. Synthetic steroids, these get a bad rap, mainly the anabolic steroids that have been abused by athletes and different people. There are therapeutic uses for these things, controlling different things that you can look at. Different scandals in the sports world, maybe you've heard of Balco and Barry Bonds, and there's all sorts of laws about these things. But the birth control pill is also a synthetic steroid. It has an alkyne on the D ring here. It's a progesterone agonist. Prednisone for inflammation. Some of you have probably taken prednisone. In fact, that's one, and this pathway has been shown to have potential to help out with coronavirus infections, right? The inflammation in the lungs is mitigated by these prednisone type analogs that control inflammation. Anyway. Let's get to the lipoproteins, LDL, HDL. You've heard those terms. People use them all the time, even in the common vernacular, which is always amazing. Yeah, so these technical terms get into common everyday usage. And my LDL level's too high or too low or whatever. Well, these are serum fat transporters. Okay, these are vesicles, kind of like micelles that transport what fat and grease and stains off of different surfaces, right? But these transport what? These lipids. Okay, so we're talking about this in chapter 18. They're not water soluble, so they have to be transported in these vesicles. And these have these phospholipids where it's just a monolayer ball. So it's kind of much more like a micelle of soap. And in the interior is the fatty position, right? The exterior has some proteins in this membrane, in this monolipid membrane here, but there's cholesterol in there, and there's cholesterol esters and acyl triglycerides. Now the LDLs tend to be the bad ones. They're bigger, okay? And they can deposit out in the interior of different blood vessels, arteries there to give disease there. They're higher amounts of cholesterol. So yeah, if the diet and the liver is pumping out too much cholesterol, you'll tend to have too many LDLs in the body and too little protein that's on the surface. That's a marker for where these are transported to. So these need to get to different positions around the body. And they're delivered to membranes as well. Cholesterol, like I said, is part of the component of all cell membranes. So it has to be delivered, transported through the blood, the serum, to get there, right? The higher density ones here are smaller. These are the good cholesterol. These are the numbers that are actually reported when you do the blood workup in the clinic, right? Bad cholesterol, in the 150 to 200 milligram per deciliter range. Kind of a funny unit there. Ask a clinician sometimes, what are the units on those LDL, HDL units? And none of them know. I torment them when I ask about it when I go to the doctor, and yeah, they should know that, but nanomolar amounts, that's tiny amounts. It's not much in the serum, but yeah, if you get more here, and it's too big, too much cholesterol in there. They have these larger shapes that are amorphous. This is a diagram of an HDL. They're more spherical, more compact, and smaller. A lot more protein is dominant in the structure than the cholesterol. So that tends to what? Make them more water soluble, right? So that kind of comes out there. All right, also mentioned this. Here's that key step again that we saw, HMG-CoA reductase, reducing the thioester down to the primary alcohol. These drugs, the statin drugs, lovastatin, simvastatin, and Lipitor, have made these companies and shareholders billions of dollars over here. Lipitor for years was the highest selling drug. Maybe it still is. I don't know. There's many other newer statins now. And what do they inhibit? They inhibit HMG-CoA reductase, and they limit the amount of cholesterol produced in the body. So this will dramatically lower the number of what LDLs the person has in the body. And that correlates with a better outcome as far as heart disease and cancer and a number of other conditions. Too many fats, too much cholesterol in the body and being deposited in the wrong places can be mitigated by inhibiting the biosynthesis. Now, this isn't affecting the dietary cholesterol. You need to control that as well. The statins, the synthesis happening in the liver. I think that's enough to say on that. You can look at the structure. Oh, let's look at the structure. So what you see is a beta hydroxy lactone in this case that can ring open. Beta hydroxy acid. You see that right here, beta hydroxy acid, and right here, beta hydroxy acid. So that mimics that part and binds to that part in HMG-CoA reductase that mimics the endogenous substrate. So the drugs are molecular monkey wrench there that bind and inhibit that. Here's a blow up of Lipitor bound to the active site of HMG-CoA reductase. You see it in there, a lot of interactions between different side chains. This binds more tightly than the substrate. So yeah, it's a nanomolar inhibitor there. You can look this up if you have more interest on the protein data bank. That's the 4-letter code for this structure that organizes a lot of these structures there. I think that's all we need to say there. Very good. Let's get out of 28. So that homework is due tonight. And let's look at metabolism. So what do we have here? Again, this is an extra topic. Some of these reactions are mentioned in your book. I've pointed them out at a couple of stages when we did the Aldol and the Claisen reaction. We'll see those again, but metabolism in its entirety is not covered in your book. It is in the supplement, the McMurray book that you can get in the bookstore. I've talked to a couple people who have gotten that. You don't need it. Everything you need to know for our class is on my overheads here, which are, of course, on Learning Suite. So metabolism is really 3 things here. We're talking about metabolizing or converting glucose into energy. That's what this means, right? That's 3 stages: glycolysis in the cytosol, glucose to pyruvate. You've already seen a little bit about pyruvate to acetyl-CoA. That's the linkage point. Now we're inside the mitochondria doing the citric acid cycle, which then decarboxylates those CO2. There's a combustion product, and that's all metabolism is. We're burning glucose. Well, there's no flame, but there are the combustion products, CO2 and water. And we are using oxygen. It only shows up at the very end, electron transport. And that's where the 2 reduced cofactors, NADH and FADH2, are converted back to NAD plus and FAD. And these electrons and those hydrogens are transferred to oxygen to make water. Yes, water is made during glycolysis. CO2 is also made. That has to get back through the bloodstream. That's a gas, right? That's converted or hydrated to carbonate. You just saw carbonic acid or bicarbonate, right? And that has to be transported back to the lungs and expelled or breathed out. We breathe in oxygen to facilitate metabolism, the conversion here. And where's the energy at? Well, it's ATP, adenosine triphosphate. That's the main energy molecule within each cell, in particular muscle cells. It has to phosphorylate myosin and actin, which gives the mechanical contraction of muscles to let you move. There's a lot of other usages of ATP, though, in the body, including in the initial stages of metabolism, up here, glycolysis, that we'll see. So we'll look at it. There's 10 steps in glycolysis, 8 in citric acid cycle. I'll show you electron transport. I don't expect you to know the mechanisms or anything there. But here again, like I said, I'm not expecting you to memorize these pathways. You've probably seen them in other classes. I'm expecting you to inspect each individual step and say something about what product arises given a condition and an enzyme. And the enzyme often tells you what's going on, or given the product, you know, what was the starting point. So inspect an individual step within the reactions, okay? So not the whole thing. Here's the important cofactors. We've been talking about these. Here's NAD plus in all its glory. This is the part we've just been looking at, and that's where the mechanism occurs, the pyridinium warhead part of the molecule. Here's the adenosine part, nicotinamide diadenine dinucleotide. It is a diphosphate. Okay, NAD plus to NADH. Here's FAD, which is a flavin, a tricyclic structure. Functions the same thing. It is an oxidizing agent. Here's the reduced form of it, FAD H2, with 2 more hydrogens here and more electrons. Okay. That can also reduce things, and like I said, that reduces oxygen to water later on, we'll see. Acyl carrier protein, we've been doing this also. Bioester right here. Here's the business end, that 2-carbon unit. That's it. All the rest of this goes along for the ride. The pantothenic acid with the thiol here on the side, the diphosphate, and what? Another adenosine. And these are oxyribose, right? These are RNA type nucleotides. There's an OH there at the 2 position. If that wasn't there, that would be DNA, okay? So RNA molecules are highly dominant in the cell. Remember we talked about transcription and translation last time. There were many types of RNA molecules, right? Messenger RNA, tRNA, and rRNA, ribosomal RNA. So you could view these cofactors as just variations of those RNA nucleotides, but they have specific groups on these positions that do different things, we can say. All right, glycolysis. We'll show the overall pathway here, and what you notice is glucose up here in the Hayworth depiction. Molecule you know and love, right, left, right, stereocenters, all equatorial in the chair, right? Going to pyruvate, okay? This happens in the cytosol. There are 10 steps. I've got them all numbered here. And what you see is first step, hexokinase. A kinase puts on a phosphate. Where does it get the phosphate? From ATP. I'm not going to go through that mechanism. We'll show you a little bit about it. Now, we won't go into the energies of things, but look at the name of the enzyme, hexokinase. Okay, hexo, 6 carbon. That actually puts phosphates on a lot of other monosaccharides, but the important one inside of a cell is this. In fact, we ought to say something about how did glucose get into the cell in the first place? Right, it needed the signaling molecule insulin to prompt the glucose transporter to bring in glucose from the bloodstream. Remember, glucose is also called blood sugar, and it's what? Not charged, but once it gets inside of a cell, it is charged now. Hexokinase puts on a phosphate. That phosphate has a what? -2 charge. So now it can't partition back out through the membrane. Remember, the membrane has a nonpolar region on its own. Now this is marked to stay within this cell. So this phosphate group has a couple different roles we'll talk about here. Isomerase, we've talked about this too. We go from glue to fru. What's fru? Fructose. This is the ketose, and there it is at the 2 position. That's a ketone. This is the furanose form, and so we're just isomerase. In fact, we've already done step 2. I showed you isomerase and how that works, right? It does the enolization and then reprotonates on the other side. It's a tautomerization process. And then it snaps back shut. That isomerization goes via the open form, and this is a hemiacetal. Glucose is in equilibrium with its ring open form, the free aldehyde. And one of the things it can do there is isomerize over to fructose, okay? Now, if you eat dietary fructose, it's sweeter than glucose, and you skip these first 2 steps, you get into metabolism quicker. And if this builds up, this can actually be converted into fat. We won't get into that, but you'll see later on in biochemistry how all these pathways of the different biomolecules, amino acids, fats, and sugars can be all interconnected. But the main energy molecule is glucose. So glutafru isomerase, and then another kinase, fructokinase, which puts on another phosphate group. So at the 6 position and the 1 position on fructose, we have there. So what's the overall charge of this fructose diphosphate? Yeah, -4. Okay. So know what these inorganic phosphates are. I'll go to the board and show you that in a second. Here's our first key step we need to go over. It's an enzyme called aldolase, which means what? It does the aldol reaction. Well, it doesn't actually add another enolate here. It actually does the reverse. It's a retroaldolase. It's called aldolase because in the forward direction, it can do the aldol with DAP and GAP, these two products, and form more glucose. This is the gluconeogenesis pathway you'll see later on. It uses that same enzyme. Okay, so knowing the enzymes, putting in some effort to learn what the names mean and what the products mean is important there. The 2 molecules from breaking this up and doing a retroaldol is DAP, which is dihydroxy acetone phosphate. So the A stands for acetone. Where's acetone? Well, it's right there. It's the 3-carbon ketone, but there's a phosphate on it, okay? Dihydroxyacetone phosphate, DAP. And then we got GAP, which is glyceraldehyde phosphate. Here's glyceraldehyde that we've already learned, but at this position, it's got a phosphate. And that came from the phosphorylate fructose, so we'll need to look at that. Here's a step with NAD+. It's a dehydrogenase. This is glyceraldehyde phosphate dehydrogenase using NAD+ as a cofactor. It goes to glycerate, which has the glyceric acid phosphorylated right here. So that's an aldonic acid. We talked about oxidizing carboxylic acid. So we'll see how to do that. We'll do a little bit on that. I think that's the trickiest mechanism right there. It has the cofactor here. Then we finally get out a molecule of ATP. ADP, diphosphate, can grab that phosphate off the glycerate phosphate position. That's exothermic. That's going downhill, making the energy molecule ATP. And then glycerate undergoes a mutation to the 2 position of the phosphate. So that's glycerate mutase. That's what that enzyme means. And then there's an enolase, which means what? Formation of the enol. Just eliminates water right there. Okay, it's elimination reaction. We've seen dehydration reactions before, right? And then this phosphate can be transferred to another ADP, and then the electrons come down, and you protonate out here at the 3 position, and that is pyruvate, the ketone right there. Okay, so let's go through some of the details of these, and then we'll go forward into a citric acid cycle. So let's go over here on the board here and look at these individual reactions now. And I'm not going to do all of them, but we'll do some of them, at least mechanistically, so we see what's going on. So this first step, here we have what? We have glucose, right? And I'm drawing in the chair form because that's more representative. 99% like this, 1% the free aldehyde, right? It is a hemiacetal right there, so you got to keep that in mind. What's the enzyme right here? Yeah, hexokinase, and that's going to phosphorylate with ATP. And I'm drawing ATP in a funny way. I'm drawing the 1 phosphate on the end, and then it's connected to ADP, diphosphate, okay? So what's the reaction here? Well, there's an enzyme. The kinase takes the proton off. That allows the electrons to come in, have an intermediate, collapse back down, and kick off what? ADP. So ADP comes off, and you get the phosphate on that 6th position of glucose. So that's 6-phosphoglucose, okay? That's step 1. And so we often just abbreviate that as PI, or inorganic phosphate, on that position, but you should see what it is. And what's the charge there? The pKa's here are 3 and 1, or no, 2 and 5. So at pH 7, these are ionized, right? So we've got double negative charge. That cannot get out through the membrane now. It stays in there. And this is the open form of it. So we're just doing equilibrium there. And step 2 is what? This isomerase, right? Which is isomerizing from glucose and putting the ketone here. Remember we did that? We took the protons off here and then we flowed them I'm sorry, they took the proton off here on the other side. Sorry. Okay. And then protonate up there, and that give us the enol, remember? I think we can review a little of that. So we got the enol form and the rest of it there. And then how do we get the ketone there? Well, we've got to take the protons off there and then flow them back in and then protonate at the 1 position. So the isomerase step, and we did that before, right? So that's now fructose. Then the next step is to put a phosphate group on that 1 position. So now we have the diphosphofructose, okay? And I'm showing the open form. In the chart there, I had it cyclic. So this is, you know, the 1% or whatever in the open form. And that's how the aldolase works. So here's our first step to really go over in some detail. And it is an aldol reaction, but it's the reverse of it. And so what we're going to do is grab this proton off the 4 position here, and then we can flow these electrons in here, right? And then break these electrons right here, and then what? Protonate up here, okay? But if we do that, we end up breaking that bond right between the 3 and the 4 position, because here is the aldol products, right? The 4 hydroxy and the ketone at the 2 position. So they are beta to each other there, right? So this is the reverse of the aldol we've seen before. And the initial intermediates here is one of the products, glyceraldehyde phosphate, or GAP. And there it is right there. That was our 4 position in the fructose. And now we have the enol form here of DAP, or the dihydroxyacetone phosphate. And so how do we get to this? Well, we've got to take that proton off here, flow the electrons back down, and then protonate right there, okay? And that will give us the primary alcohol, or DAP there. The next step is an isomerase of DAP into GAP, okay? It's a different isomerase than the glucose isomerase. It's a 3-carbon triose isomerase, but that will give us effectively what? 2 equivalents of GAP, okay? And this is kind of key thing. We have 6 carbons here in glucose and fructose, and now we have the 3 carbon units, GAP and DAP, okay? But then we isomerize DAP to GAP there to give that. All right, let's look at the glyceraldehyde dehydrogenase. And that's kind of a key one there, I think. We're going to take this molecule, which is our GAP, okay? And it is chiral, of course, still has the stereocenter right there. And we're going to react it with NAD plus, okay? And the enzyme is glyceraldehyde dehydrogenase. And this one, I think, is a little unfortunate because it doesn't really say much about how dehydrogenase. It sounds like we're taking off hydrogen there, but you know, we're putting a phosphate on there, and we're actually using inorganic phosphate here as part of the things to get this on here. And you know, dehydrogenase, that's kind of unfortunate because, well, there's 1 hydrogen that's removed there. I guess that's part of it. But with dehydrogenase, you think of minus, you know, 2 hydrogens going from an alkane to an alkene, whatever. So this name's a little bit unfortunate, but certainly it does oxidize, right? It goes to the glycerate phosphate ester there, okay, from an aldehyde. So we are doing oxidations here, and that's all of metabolism is. We're eventually getting to CO2. So you can follow these pathways throughout and see that we're, you know, putting more oxygens on or taking hydrogens off. So this is kind of a key step, that way of doing that. So how does this work? Well, in the enzyme, there's a thiol and a base that can take off that. It's a cysteine. It takes off the hydrogen there and delivers that there and gives a proton up here, okay? So let's look at that intermediate. We're adding a sulfur as a nucleophile to the carbonyl. And that's okay. This is electrophilic, right? Carbonyls are electrophilic on carbon. So, you know, it is a concerted pathway and a lot of arrows there, but you can see we're going to protonate up there. Let's see what the intermediate looks like. So the squiggle means, you know, the rest of the enzyme. I'm not dealing with that. And then the rest here, the phosphate and the alcohol. So there it is, tetrahedral intermediate, right? So it's similar to that regard. Now we use the NAD plus, and we'll just draw the business end of it, the nicotinamide part. Don't forget about the plus charge, and I like that about NAD plus cofactor. They remind you of what that formal charge is, right? And so what goes on next? Well, the enzyme has this base, still there. Actually, we just generated up here, right? Can take that proton off, flow the electrons in here, and flow the electrons with the hydrogen in here. Okay, that will give us NADH coming off, okay, which is the reduced part of the cofactor after it does its oxidation. And what? The sulfur now ester. Okay. And what? We've got the thing oxidized. It's now carboxylic acid oxidation state, right? A thioester. How do we get to the phosphate ester? Well, we've got inorganic phosphate. Not drawing out the whole phosphate unit, but we can look at the tetrahedral intermediate if you'd like. We get the sulfur, O minus, with the phosphate on there. And we get the rest of the molecule here. And what? This can come back down, and off comes your sulfur. And that can grab a proton on the enzyme, right? And give you back the enzyme. The enzyme has to be regenerated. And what? That gives you the glycerate with the phosphate on there, okay? Let's see, we'll skip ahead. The mutase just transfers one phosphate up to another point. Let's look at the enolase now. And yeah, so this is step 9. Let's look at the last couple of steps here of glycolysis. So we've got this. After that phosphate comes off onto ADP, we get this step. So this is step 9, if you're keeping track there. And you can go back and look at the chart there. And the enzyme here is called enolase, and it loses water. So given these conditions, you know, where can you lose water here? Well, just right here, okay? So let's see. Oh, sorry. This is actually at the 2 position phosphate now. Yeah, just got to double check that. And what we're going to get is this. It's ionized there. Phosphate at the 2 position, and then that. Okay, so there's the enol form that's phosphorylated. So how does this work? Well, the enzyme has a base, and there's the proton we need to take off to form water, right? And how can the water just come off? Well, there's a protonated side chain here. Now, these are usually imidazoles and imidazolium groups. Sometimes lysines are involved here. What do? We take the proton off, flow the electrons in, and break these electrons off. Now, OH minus isn't coming off as a leaving group. In fact, you could imagine protonating that first. It's close to that side chain. It's known to be concerted, happening very close together. But it is an elimination reaction, okay? But it happens all at once. There's no free carbocation here. And this is not a hydroxyl as a leaving group. It's coming off as water as it's nearby, proximity-wise, to this protonated side chain in the active site of the enzyme to give the eliminated product, the enol product, okay? So that's 9. How do we get to pyruvate? Well, here's pyruvate, which has the carboxylate and the ketone there. So what do we need to do? Well, this actually is called pyruvate kinase. And kinases do what? They transfer phosphates. And where are we transferring a phosphate? Well, we're transferring a phosphate from ADP, which is the adenosine diphosphate here. It's grabbing this phosphate here, And let's leave these electrons on the oxygen. So we're doing part of the mechanism here. That will give us this. And plus ATP. Okay. So there's where the ATP forms. The diphosphate adds to that phosphate 2 position to give us this. And what's that enolate? Well, that's just, right? Flow the electrons down here. And in the enzyme, what? Grab a proton at that position. That gives you that methyl group. Okay. So there's pyruvate. And I think those are the key steps. The glyceraldehyde dehydrogenase is certainly the toughest one, the unique mechanism. It's a variation of the NAD+. The aldolase is unique, I think, from what we've seen before. It's a retro-aldol. In fact, we ought to say a little bit more about that. Maybe we went a little too fast on that. Let's look back at our aldolase here. Why does this occur? You're breaking a carbon-carbon bond. That's worth about, what, 90 kcals per mole? Why is this favored here? In fact, it depends on the conditions the organism is under, that the cell's experiencing, whether it needs glucose or needs the energy from glucose. This same enzyme will do gluconeogenesis or glycolysis, okay? And what affects this equilibrium? Well, breaking this bond is certainly a very high barrier, right? 90 kcals per mole. But what is this charge? Overall, this charge is -4. Ouch. To have that many like charges in a small molecule like this, that really weakens this bond electrostatically. So this is actually not 90 kcals per mole energy-wise anymore. It's actually down around 40 kcals per mole. So this is certainly breakable with an enzyme. And if the organism, the cell, needs the energy, this can be favored then equilibrium-wise. You'll learn all about the control about it later on. I'm not going into that, but you see each one of these small ones now are negatively 2 charged, okay? And that allows them to be solvated and to mitigate this -4 charge within that molecule. So the charges are actually important. The phosphate groups, they seem like it's kind of willy-nilly. A phosphate goes on here, comes off there. Well, there's a process for this. And you'll appreciate the overall scheme later. What I want you to focus on now for our class is just use what we know to think about things like dehydration, which we've seen before, enolate formation, protonating and enolate, that kind of thing, okay? So use what you can about the conditions and what the result is to think about a specific mechanism. But don't try to memorize the overall pathway and all the control issues that go on there. Okay, let's go back up to the overhead here and see what else we need to know. We still have a little time here. We might have to finish metabolism next time, but yeah, there's glycolysis and all the stuff. Yeah, look back through that and just know the basics of that. On a quiz or a test, we could give you, like I said, the starting material and the conditions, the name of the enzyme and the product, okay? Or if we give you the starting material and the product, then you should be able to come up with something about the conditions or even the name of the enzyme. And if you're kind of close to the name of the enzyme, we'll give you full credit, okay? Like I said, some of them are kind of a funny name, like glyceraldehyde phosphate dehydrogenase. Well, that does an oxidation of this aldehyde to the glycerate, carboxylic acid phosphate. But the others are pretty straightforward. I think enolase, mutase. Pyruvate kinase, that's kind of funny too. The phosphates transfer from ADP to ATP, and then you protonate to get the methyl group there. Anyway, okay. So we've got pyruvate, and we've already seen this graphic actually. I showed you pyruvate decarboxylase. Has the 2 cofactors, thiamine, and what's the other one here? Oh, NAD+, there it is. And yeah, this is a tricky mechanism. I've never put it on there, but you can appreciate each step. You can understand the flow of electrons. Here's NAD+ doing the oxidation to give the thioester right there. And this is what enters then the mitochondria and then does the rest of the thing here. It's a roundabout. It's a cyclic relationship here. And this is kind of funny because, we started with pyruvate, the 3-carbon unit, right? And here's 1 molecule of CO2 being generated. So that's the first combustion molecule or product here from glucose, right? CO2. And we've created two of these 3-carbon pyruvates from one molecule of glucose, right? And don't worry about that molecular bookkeeping. They'll torment you with that in Biochem 481 later. But it falls out of the structures and the mechanisms, what we're talking about. Where are all the carbons, okay? Well, here's the 2 carbons, which originally came from glucose, right? And here's 2 more CO2s right here and here. And you go initially reaction up with oxaloacetate to citrate. That's a 6-carbon unit. Then back down to 4, and then, or back down to 5, and then down to 4. And then this is just converting succinate all the way back up to oxaloacetate. So there are some funny names here. Succinate, succinic acid, there it is. I had you learn those dicarboxylic acids. Succinyl-CoA. Fumarate is the oxidized form with an alkene there. And then hydrated form, that's got the secondary alcohol. And then oxidized, this is the alpha-keto acetate. So oxaloacetate from oxalic acid, the 2-carbon dicarboxylic acid, and then an acetate unit stuck on it. That's where that term comes from, oxaloacetate. It's kind of a funny name. And then citrate, speaking of limonene, here's another citric, citrate fruit product. Citrate is in those fruits, lemons and limes and oranges. It's also in our mitochondria as citrate, which is an aldol product from acetyl-CoA adding to the ketone here. So we'll review that. We'll look at that. And then there's an isomerization from aconitase to isocitrate. Okay. And then isocitrate dehydrogenase to alpha-ketoglutarate. And that's a decarboxylation oxidation. That's kind of a cool mechanism. I'll probably show you that one. This one right here, alpha-ketoglutarate to succinyl-CoA, that's completely analogous to pyruvate to acetyl-CoA. In fact, the mechanism is almost identical. But there's where you generate the 2 CO2s, decarboxylation there and decarboxylation there. Okay, and the rest is just cycling up to accept more acetyl-CoA from the cytosol. Okay, so it is a roundabout relationship here. Cyclic in that the final product is the precursor for the next round, you could say. You see the ATPs here in blue. Those are generated after electron transport chain inside the matrix of the mitochondria. So there's a couple things anatomy-wise we need to mention about places within the cell. But that's after electron transport chain. And that's, you see NADH, NADH, and FADH2, which are fully converted into ATP and generating water on those last steps. So you don't see those yet. Those are other steps we got to do there. So yeah, let's see what we need to know there. Oh, electron transport chain, we'll get to that. Let's do a couple mechanisms here, the last couple minutes that we've got. So citrate and yeah, and then the oxidation at the end. So yeah, 8 more reactions. So yeah, we're really tormenting you here, I know. But again, part of this is getting you ready for biochem and to show you how the reactions you've already learned relate to these biochemical pathways. So the first step here is taking acetyl-CoA, which is the thioester, and then reacting it with oxaloacetate. And that's kind of that funny term there. And yeah, I'll keep it in the Fisher projection. That kind of saves some space there. And we're doing citrate synthase. And so that tells you what we're making, right? We're making citric acid here. And what's citric acid look like? Well, it's a dicarboxylic acid that's ionized. It's got methylenes here and here, and another carboxylate there, another methylene, right? So yeah, it's got how many carbons? 1, 2, 3, 4, 5, 6 carbons. And here's the 2 carbons coming from glucose, okay? So how does this work? How do we make citric acid, 3 carboxylic acids, and an alcohol, right? It is a tertiary alcohol, so at least you can see that part. And the oxaloacetate part is what? Right here. So we're going to make this bond right there, okay? So let's see, how are we going to do that? Well, the enzyme has a base, and that base can grab that proton. It's stabilized as an enolate. It doesn't form a free enolate. It's in proximity with the oxaloacetate. So this can come in here And then there's a protonated form of the enzyme. So yeah, this whole thing is the enzyme, sorry. So active site has both substrates in there. And when it does that, it forms the aldol product. So let's just redraw it here with that on there. And yeah, we've got that kind of twisted around there, CoA. And the other methylene and the other carboxylate. Okay, there it is. Yeah, what else do we need to do? Oh, we need to add water here. And the enzyme can do that. And that's just what? Tetrahedral-mediated hydrolysis, proton transfer. And that's the carboxylate up here. So here's our 2-carbon unit here from acetyl-CoA. And there's where our oxaloacetate was, the citrate. Okay. So what type of reaction is this? Well, it's an enolate adding to a carbonyl. So what is that? That's an aldol. Okay. That's perfectly analogous to that. And the product here is what? This beta position. Here's the alpha position to the carbonyl, right? So yeah, we could make that from an aldol. So look at the similarities and that variation there. All right, let's do the next couple of steps here. We got some time. Step 2, and maybe I'll redraw the citric acid here a little bit different. So here it is, yeah. So you see those methylenes are kind of kinked down there now. Aconitase is the name of the enzyme, and that's kind of unfortunate because you don't see anything named aconitase. You see citrate, and then you see isocitrate. And isocitrate is this, where the hydroxyl group is just moved over, right? So here's isocitrate. So citric acid to isocitrate with aconitase. What's aconitase? Let's see. Well, the enzyme does this. The enzyme does a dehydration first, and it also has a protonated base that can grab that and form water. Let's have water coming off. And what do we generate then? We generate the alkene right there. This is actually aconitic acid, and that was known before this step in the mechanism of citric acid cycle was fully developed by Krebs and others. A lot of history about the citric acid cycle, all the intermediates being figured out. Some people say, well, this should be a 9th step. That should be a 9th intermediate. Well, no, it stays associated with the enzyme. It never leaves. So it's never in the aqueous solution there. What do we need to do to go back here? Right, we need to add water. Okay. So how does the enzyme do that? Well, here's water. How can we do that? Well, we can take off proton. That will allow hydroxide to attack there, right? And then we need to protonate with more right there. And that gives us the isomeric form there. Now let's look at these 2 alcohols. What type of an alcohol is this? Right, it's secondary alcohol. What type of an alcohol is it in citrate? It's a tertiary alcohol, okay? We need to do an oxidation decarboxylation. And in order to do that, we need the secondary alcohol, okay? So this functions allowing us to do an oxidation. Remember, we said tertiary alcohols cannot be oxidized. So remember, this is an oxidative process, and this isomerization, aconitase, sorry, the name of the enzymes kind of masks what's going on. It's kind of a historical thing relative to this acid, this triacid, which was known on its own, but never dissociates from the enzyme. And it just does dehydration followed by rehydration to isomerize that position now. Okay. So yeah, I think that's all I need to say there. Let's look at the next step, and I think this will be our last topic today. Yeah, we have a minute here. So let's look at the next one, which is this one, isocitrate. Step 3 here, which is going to go to alpha ketoglutarate. And this is kind of a key thing too, because we have 6 carbons here, right? And now we're going down to how many? 1, 2, 3, 4, 5. Yeah, 5 carbons, okay? And let's see, what do we need here? This is alpha keto glutarate. Oh, glutarate, glutaric acid. There's another dicarboxylic acid, right? 1, 2, 3, 4, 5. There's glutaric acid. Glutamic acid is the amino acid, right? If there was an alpha amino group there, it would be glutamate, right? But this is glutarate, which has the 5 carbons there, and then alpha keto. So alpha keto glutarate, and that's the product here. And it uses NAD, okay, to do this oxidation here. So what's going to go on here? How are we going to do this reaction? Let's see. So the decarboxylation, alpha keto glutarate decarboxylase, how are we going to do this? Well, let's see. We've got a hydrogen here, and we've got to get rid of CO2 right here, and we're going to do an oxidation right here. In fact, let's do that first. We are using NAD plus, right? So we've got the plus form here. Sorry. Got the amide right there. And let's see. So our enzyme is going to have a base that can take that proton off, flow the electrons here to form the ketone, and then flow these electrons in and push them around and do the reduction of NAD+, just like we've seen before, right? So that will give us this, which is now a ketone at that alpha position. And got the other one there, right? Plus NADH, okay? So there we go. But what about this carboxylate up here? Now I'm drawing that one at the 3 position in its full form there to accentuate that. So we just did the ketone formation, right? The oxidation, NAD+. Now how do we decarboxylate at that 3 position? This is the key thing here, right? So we can allow for this to form, right? Protonate here. It's beta keto carboxylate, and that's just like the malonate and the acetoacetate decarboxylation we saw before. But now we're under physiological conditions. We don't have the neutral protonated carboxylic acid. We have the carboxylate. But you can see we're what? We're beta to this other carbonyl here. So we can leave behind this pair of electrons right here and protonate there. And that will give us the enol form, right? Of that, yeah, plus CO2 just came off, okay? So yeah, we've got this, and you know, we've been talking about enols and enolates and tautomerization. That's our last step there. But you see, this can weaken this bond and allow for the decarboxylation. In fact, you know, this loss of CO2, that's our second spot where we're forming a combustion molecule, right, CO2. And we're down to the 5 carbon unit now. How do we get this from the enol to the keto version? Well, the enzyme, again, just has a protonated form there and base that can take off the proton, flow the electrons, and then protonate and give yourself the product there. Okay. Yeah. Let's see. Let's go back up to the chart here to wrap up with. And yeah, we're out of time, but we've gotten a lot of progress-wise, right? We've gotten down here to the alpha keto glutarate using the Isocitrate Dehydrogenase, forming the alpha keto product. Sorry, I got the name wrong there, I think, on the board there. Yeah, this is the Isocitrate Dehydrogenase, this first one here. We'll look at this one next time. Well, we won't go through the details of that. We've seen it already. It's got the thiamine cofactor. It's analogous to the pyruvate to acetyl-CoA formation there up at the top. But then we'll do the succinyl all the way back up to oxaloacetate, and you'll see the rest. And we will, you know, wrap up with electron transport, show you a little bit about the anatomy and everything, and I think that's it. So yeah, we'll be able to finish this very quickly on Monday, and we'll be reviewing, of course, for test 4 and for the final on Monday. We'll have the 2 hours on Monday to go through that. But be reading, of course, and looking through the sample materials. So yeah, test 4 is just these 3 chapters, primarily 26, 27. 28, not much with the lipids, right? And then metabolism, just individual reactions, and maybe just a few on the test, okay? So not a lot. The first 2 chapters are the important ones there. So yeah, keep this straight, and we'll see you on Monday. Very good.